Welcome back everyone. In our last session, we navigated the basics and where to find dataset on NCBI, Geo and Array Express, a quick overview on microarray techniques and the journal roadmap for gene expression analysis. So I hope you all have your dataset to practice with because we are going to start the actual analysis from this session. Specifically today we will focus on pre-processing of microarray data in R using bioconductor packages. So before we write any code, I want to quickly talk about bioconductor. This is the home page of Bioconductor. Basically, Bioconductor is an open source project that provides thousands of R packages specifically for bioinformatics and computational biology. So at this time, Bioconductor has more than 2000 packages, 2341 R packages at this time. That covering almost every type of omics data analysis, microarray, RNA sequencing, single cell analysis, proteomics, and you name it. Smart about bioconductor is that these packages are developed and maintained by scientific community, and these packages almost always come with great tutorials or reference manuals and also with working examples. And if there is something that doesn't make sense to you or you get stuck at any point of your analysis pipeline, the bioconductor support site is a great place to ask questions or find answers. So this is a fairly active forum, a really nice interactive community where queries are addressed by the people who already have encountered the issues, coding errors through their own work. To learn about bioconductor, there are plenty of learning materials available. If you have a time, review them. You will find lots of learning resources there. And these bioconductor packages are divided or organized into a different categories. For example, softwares, assay data, experimental data, annotation, etc. About gene expression, there are 607 packages available at this time that address the gene expression data analysis in a different ways. You can search for package of your interest from these categories. For example, Array Express is a package that lets you access dataset directly from Array Express Biostudies database. You already know how to fetch a dataset from Array Express manually, but if you want to fetch data directly into R, you can use Bioconductor Array Express package. To work with the Bioconductor packages, the first thing you need to do is to install BioC Manager. As the name suggests, this allows to manage bioconductor environment in R. It helps you install and manage bioconductor packages. This is actually come from CRAN repository or not from bioconductor, so you can install it by using install.package function in the similar way we use to install any CRAN package. After installing BioC Manager, you can install any bioconductor package using BioC Manager colon install function, and in this process make sure the version of package you are using compatible with each other and with your version of R. So this Array Express package is compatible with R version 4.5. If you are using old version, you can check out here. Here in this Array Express package, you can find out all the relevant information or required codes you need to download Array Express data in R. If you look at the end of this Array Express package, you will find the example of microarray data analysis workflow that starts from fetching microarray data from Array Express. Then they perform normalization and explain you how you can conduct differential gene expression analysis using different bioconductor packages. Similarly, we will use some other bioconductor packages throughout. Now let's talk about the dataset. The specific dataset for today is from GEO database with the session ID GSC 79973. It contains Affymetrix microarray data comparing 10 tumor tissues with 10 adjacent normal tissues. I have already downloaded raw data files. Also, we will need this series metric file as well. We will directly download in R. I will show you in a while. You can also fetch raw data files in R directly using bioconductor GEO query package. I will show you code, but sometimes this automatic download fails if you have a weak internet connection. So that's why I recommended you in last session to download raw data manually. Now let's move to RStudio and start our work. The first thing we need to install some bioconductor packages and CRAN packages. Since you are using bioconductor for the first time, the first thing you need to install this BioC Manager package. After the installation of BioC Manager, you can install these bioconductor packages. And to load these packages, we will use the library command for all of these packages. So these are the some packages we will use in this session. The first one is GeoQuery. This package allows us to download dataset from Gene Expression Omnibus GEO. We can retrieve both process or raw files and series matrix file, which are essential for pre-processing. Next, we have FE package that we will use for pre-processing and normalization of microarray data. Then we will use array quality metric to check the quality of microarray data. Next, we will use these CRAN packages for data manipulation. So load all these packages. Now, the first step is to download a series matrix file. So to download series matrix file, we will use the function get GEO from GEO query package. The first argument is the session ID of the data, that is GSC 79973. You can replace session ID with your data. The next argument is GSC matrix equals to true. And to save this series matrix data, let's create a variable GSC data. If I run this line of a command, I will start fetching the data from NCBIGU. For this step, you need internet connection. Here, this argument GSC matrix equals to true tells the function to return a series matrix object which contain process expression data along with phenotypic and feature data. We will not use the process data. This is just to show you how you can download process data that are already normalized. In this course, we will work with raw data file. So we have the GSC data files here. Here you can see experimental data. Here you can find the lot of information. This is the actual expression data, or you can say it's a process data. Next, we have a phenotype data that contain all the information regarding the samples, metadata or clinical information of these samples. Next, we have feature data that contain annotation, gene samples and gene names. So to extract these files, we have different commands. For example, if you want to fetch expression data, we can use the command express, then the file name GSC data, selected GSC matrix. So this expression function extract the expression value from the GEO object. Let's save it into expression data variable. So these are the normalized process expression data set. In the similar way, we can extract feature data by using fdata function. This command will remain same, but this fdata will fetch the feature data from this GSC, save it into feature data variable and run this line of a command. So we have feature data with 16 columns that contain probe IDs, the species, annotation date, experimental type and other information. Column of interest for us in this feature data is these gene sample or entries IDs. We also want to see the phenotype data, which tells us about the each sample, like tissue type, condition, treatment or metadata. So to extract phenotype data, we have a command pdata phenodata. Again, the argument will remain same, and let's save it into phenotype data variable and run this line of a command. So this phenotype data contain different information of this particular data. Source name column is our target column. This is very important because we will use it later to define a groups for differential gene expression analysis. Remember, having the series matrix helps you understand the data set, specifically this phenotypical data. This helps you plan your pre-processing and check for any inconsistencies or missing metadata. So before working with raw data, let's check phenotype data have any missing information or not. So we will use is.na function, sum is.na of phenotype data, and check the missing values in our target column, that is source name. So in this phenotypical data, we don't have any missing values, so we can proceed. So this gives you context about the how geo datasets are structured and what information is available before starting. any analysis. Now, after exploring phenotype data, we can proceed to downloading and reading raw data. You can download the raw data file by using get geo supplementary files from the GeoQuery package. Here, by specifying the session ID of a NCBI Geo dataset, let's say GSC 79973, by default it will download the dataset into your current working directory. But if you want to download it in a specific folder, you can specify it by using base directory function, let's say raw data, and make directory equals to true. As I have already downloaded the data manually and saved it in my raw data folder here, so I am skipping this download step. But if you want, you can use this code to fetch it directly from NCBI Geo. The downloaded cell files usually come in a .r archive, so we need to untar them into a folder so R can read each file individually. Untar them using untar function raw data file name GSC 79973 underscore raw dot jar. Then the next argument is external directory. This is the folder where all these files will be stored. So I want to store all these cell files into my raw data, and I want to create a subfolder within that folder with name cell underscore files. If I run this line of a command, a new folder has been created with all these .cell files. Similarly, if you have your dataset in .zip archive, you can use unzip command. This command works same to the untar function. Like here, I have downloaded this dataset from ArrayExpress. So to extract file from this folder, I can use this unzip function. So in this way, if you have downloaded dataset from ArrayExpress, you can follow the same steps to proceed with that. To extract metadata information, you can download strf files from ArrayExpress. So workflow will remain same if you are using Affymetrix microarray data. So we can read these cell files into the R by using Affy package. We will use read Affy function. So here in this command, the only thing you need to mention the cell file path, that is raw data, and within that raw data we have subfolder with name cell underscore files. Let's save this Affy object into a variable raw data. This read Affy function reads all the cell files and create an Affy object. So this object contains the raw intensity data for all samples. If I run this raw data, so here R will show basic information about the data, the size of array, platform, number of sample, number of gene, and annotation. So this raw data is our starting point for pre-processing. Now a quick note about the platform. In this working example, we are using Affymetrix Bioinformatics platform. If your dataset comes from a different platform such as Illumina or Agilent, you may need additional bioconductor packages to read and pre-process the data. For example, you need Lumi package for Illumina. So while following this tutorial step by step, make sure you are working with Affymetrix dataset. Now that we have raw data files into R, the next step is to check the quality of our raw data. This is very important because quality control helps us identify many potential problems with the samples before we proceed to further analysis. There are multiple ways to check the quality of microarray dataset. For example, you can create box plots to look at the intensity distribution across samples. Another method is MA plots to see the relationship between log ratios and average intensities of the probes, or another method is density plot to inspect the overall signal distribution. Instead of creating each of these plots separately, we will use array quality package. This package combines all these methods in a single QC workflow and generate a detailed HTML report. It is very handy because it is also flags potential outliers automatically, so you don't have to inspect each plot manually. And remember, this array quality metric package is specifically for microarray data. If you're dealing with RNA sequencing data or any other data, you have to use respective maps. So for this, we will use the function array quality matrix from the array quality matrix package. And the first argument is expression set equals to raw data that tells the function which data to analyze. Here it's the raw cell files we read using readFE function. Next we have out directory argument that specify where the QC report will be saved. In this case, I will create a folder called QC raw data in my results folder. So the path will be results slash QC underscore raw underscore data. Next, we have to mention another argument that is force equals to true. This option allows the function to overwrite any existing report in that folder. Now the important part is this do dot log transform argument that will be true. So why we are using this log? Because microarray intensity values are usually skewed because of hybridization intensity can span several orders of magnitude. So log transformation helps in many ways. It reduces the skewness and stabilize the variance across the genes or transcripts and probes. Without log transformation, high intensity probes can dominate the analysis and obscure the pattern in a lower intensity probe. Also, it makes MA plot and box plot easier to interpret because differences in intensities become more proportional. And most important, mostly downstream methods including normalization and statistical testing assume that data is roughly on log scale. So by setting this log transform equals to true, array quality matrix automatically log transform your data before generating QC plot. So when you run this, QC raw data folder has been created. So now R will generate HTML reports and the plots in this QC raw data folder. So you can see it created some files here. So here you can see it generated this HTML index.html file. This is the QC report along these separate PDF and PNG format plots. You can use these plots. This is useful if you want to include the QC plot in your report presentation or just to save them for a future. So if I open this HTML file in a browser, you can see this is the QC report. So in this report, this is the complete summary of a QC report for raw data. We are looking for the outlier in this step. Outliers are the samples that behave very differently, may indicate technical issues or problem during hybridization. So here is an important point. If all QC methods or most of these methods declare any sample as an outlier, we will exclude them. But we will exclude samples after the normalization step. We will not remove samples from raw data. This is the initial QC report. Here, 5 samples are flagged as an outlier by using different methods. The first method is outlier detection by distance between arrays. The next number 2 method is box plot. Then we have relative log expression analysis for raw data, normalize unscaled error, MA plot, and special distribution. Collectively, these methods flagged 5 samples as an outlier. However, it is important to note that we don't rely on a single QC matrix or method to declare it as an outlier. Instead, we cross-check sample using different QC plots and statistical criteria before making any final decision. For example, all of these samples are declared outliers by only any one method. Also, this report is based on raw data, so at this stage, outlier may be influenced by technical variation, background noise, or intensity differences. So, the final assessment of outlier will be made after data normalization. And now that we have checked the raw data, the next step is normalization. We will use RMA method, a robust multi-array average, which is one of the most commonly used normalization technique for FE matrix microarray data. So let's create a variable normalized data. We will use the function RMA function from FE package and we want to normalize this raw data file. So let me explain what happens here. The RMA function perform main 3 operations. First one is this background correction. It adjusts for non-specific signals and noise present in the raw microarray intensities. Next, it will perform quantile normalization. This makes the distribution of probe intensities across all the samples identical. So the goal is to remove technical variation between arrays while preserving their biological differences. The next step is the calculating expression. So Affymetrix array have multiple probes per gene. So RMA summarize this probe level measurement into a single expression value for each probe set or gene. Now after normalization, we need to check the quality again to make sure the data look consistent and no unexpected artifacts appear during the normalization. Since the normalized data is already log transformed, so at this step we don't need this log transform function. So I will remove this argument from this code, and instead of a QC raw data, now we will generate report for normalized data. At this time, I am not running QC for the all of the samples, but I will run only for these outlier samples. But to save time, I will only check these 5 samples. These are sample number 1, 8, 9, 11 and 20. So I need to specify these samples. So at this time, QC will run only for these samples, but you use the entire dataset to check the QC of your data. Now, a QC report has been generated for normalized data for these specific samples. If we open it in a browser, so in this report, again, sample number 8 is declared as an outlier. So this indicates that the other 4 samples were likely affected by technical variability or background noise, which we corrected during normalization step. So for the sample number 8, this discrepancy may not be necessarily represent technical outlier, as it is only indicated by the one method. And given that biological variability is expected in such dataset, maybe possibility and the sample was identified as an outlier. The discrepancy may not necessarily represent technical variability. Instead, it could reflect true biological variation, maybe difference in tumor stage. This will be a tumor samples, and that is different from other tumor tissues. We consider it appropriate to retain this sample for downstream analysis. Also, the another reason to keep this sample because we will use the same data set for machine learning application. So there we can check by using different classification and clustering methods if this sample show any variability or not. So at this time, we will not remove any sample from our data. We keep all the samples as it is and will decide based on machine learning methods. So after normalization, the next step is to extract the expression values. So let's extract normalized expression data by using expression function normalized expression, or let's say process data, because it is our process data. As the data frame expression of normalized data. Now this is our process data. Each row is a probe and each column is the sample. So in this data set, we have around 54,000 probe IDs and 20 samples. So this is the main data set we will use for further downstream analysis. But before we proceed, we want to remove probes that show a little variation across the samples. So why do we filter low variance probes? Basically, probes with almost a constant expression across all samples usually do not contribute biological information, or it may introduce noise in downstream analysis or machine learning tasks. So by removing them, we focus on genes that are more likely to be biologically relevant. To do so, the first step is to calculate the median intensity of each probe, because median is robust to extreme values and give us the sense of overall expression level for each probe. Now to inspect the median intensities, we plot histogram, plot the distribution of median intensities of probes. So by using his function, we can plot the raw underscore median, comma, breaks equals to 100, frequency will be false, and the title of the plot will be median intensity distribution. So this histogram plot helps us decide a threshold for filtering low expression or low variance probe. So here in this case, we can select the threshold. We can set the threshold as 3.5 or in between 3.4 because these are the expression values that contain very low intensities. So let's select the threshold as 3.5. We will remove all the expression value below this threshold. Threshold will be 3.5. Now let's plot a line of this threshold. So this is our cutoff. Below this threshold, we will remove all the probes. Now filter out probes above this threshold. Filter data, raw median greater than the threshold. This returns the logical vector for all of the probes. Filtered expression from process data. We want to filter data by rows. So filter data, set process data to keep only these probes. Now this filtered expression contains the probes with sufficient variation. This is our final process data that we will use for further analysis. But before moving further, look at the column names. These column names are very long and bulky. We can rename them using phenotype data. This is our phenotype data, raw names that are identical to the column names. It should be identical to the column names. To do so, we can use the function column names. We want to change the column names of filter expression with raw names of phenotype data. Now we renamed our column. So this is our processed gene expression data. I want to overwrite this as process data. So now filter expression data is ready. It contains probes with sufficient variation, clean column names, and is ready for probe to gene annotation in the next step. Now let's prepare our phenotype data. If you look at the phenotype data, you will see a multiple column, a sample title, source of the sample, organism, and other information. So for our analysis, we will use source name. We will use this source name column as our main or target column because it tells us the tissue type, whether the sample is a normal gastric mucosa or gastric adenocarcinoma. So this information we will use to create group for differential gene expression analysis for machine learning targets and for further analysis. So let's create a vector with name groups. If we check the class of phenotype data source name column, so it is a character column. For our grouping or target column, we need to convert this column into a factor. So let's create a target column with name groups, and to convert character to a factor we use this factor function. Then phenotype data, selector, source character name, levels equals to, the first level is gastric, mucosa, and the second one is gastric adenocarcinoma. For our convenience, we want to label them as normal or cancer. So in this way, we can prepare our data for further downstream analysis. In the next session, we will continue with differential gene expression.